not there in textbooks. I feel so like very little bit information is there, and ma'am will introduce all these metabolic identification topic. Although it is pharmacology, we need to learn as a part of analysis, as we have to go with metabolic identification by different types of analytical procedures. Thank you and welcome you, ma'am. Now I would like to ask Dr. Rihanna, ma'am, to introduce our speaker.
Thank you, ma'am.
Yeah.
Good morning, all. Ma'am, can I start?
Good morning, ma'am. Yeah, sure.
Okay. Good morning, all. It gives me great pleasure to introduce our honorable speaker, who had 11 years of experience in both teaching and research. Moshina Patwekar is currently an associate professor in pharmacology and toxicology at the Lokman College of Pharmacy in Gulbarga. In the year 2021, she received a Doctor of Pharmacy degree from BAMU in Aurangabad, Maharashtra. She graduated with a Master of Pharmacy in Pharmacology in 2011 from RGUHS in Bangalore and a Bachelor of Pharmacy in 2009 from Solapur University. She has achieved distinction throughout her academic career and has been a university top student in the B.Pharm course every year. More than 30 of her publications have been published in scholarly journals at the national and international level. She serves on the editorial boards of 20 reputed journals, including elsewhere publishers. She published 3 books and held 2 patents. I welcome Moshna Patwekar, madam, to start our session.
Welcome you, ma'am, and very good morning to you. Thank you, Rihanna.
Good morning, ma'am. Good morning, ma'am. I am really very glad to be the part of your webinars. I would really be thankful to the principal ma'am, that is Dr. Sunita ma'am, for giving me an opportunity. I would even like to appreciate the organizers and the team, including Rehana ma'am and Vijay Lakshmi ma'am. So with this, I think we should not waste time and I'll start my session. Ma'am,
sure.
the host needs to give me the permission to share my screen. I am unable to share it.
Oh. Pratyusha, ma'am.
Ma'am, you can share it, ma'am. I need not give any permission.
Oh, okay, ma'am. I'll just start.
Okay.
I'll just check it once again.
Yeah, ma'am. Okay.
Thank you, ma'am. Please go ahead.
Hope you all can see my slides now.
Yes, ma'am.
Yes, ma'am.
Okay, so my today's topic is metabolite identification. There are a few keywords which we have to focus on to the today's topic. Number one is metabolite profiling. What are the in vitro techniques for metabolite profiling? Human liver microsomes. Next is hepatocytes. Next is HPLC as well as LCMS and MS. So let me begin with what is metabolite profiling. I hope you might have come
Excuse
through
me, ma'am.
Yeah.
Can you make it a full screen?
Yeah,
so that,
just a second. Yeah, ma'am. Now is it visible?
Yeah, ma'am. Thank you.
Okay. So metabolite profiling is nothing but a technique where we are using the metabolites to study and as well as to develop the different drug. Okay. So we have already gone through basics like what is ADME. And considering the ADME, that is the pharmacokinetics as well as pharmacodynamic studies, we usually make a drug or we usually design a drug. So while developing a drug and designing the drug, many time it happens like we come across many metabolites. So these metabolites become a useful as well as useless part of our drug. Like they make our drug either very potent or active, or these metabolites sometimes may hinder our final product, and they lead to the drug may get lower in activity just because of the metabolites. So metabolites are nothing but the intermediates and the products of metabolism, whatever metabolism process we go through. So metabolites are nothing but the products of metabolism. Now what we are going to study is the measurement of the metabolites, where as well as their interpretation. How are we going to measure the metabolites? Like whatever metabolites we get through the production phase, those metabolites we are going to measure as well as we are going to interpret. There are various techniques through which we can measure the metabolites, and which are present in vivo as well as in vitro. So let us go with the first part, like what is the definition. Sometimes this phrase can even be called as metabolite profiling. It can even be called as characterization or identification. So what are these terminologies related to? What can we even exactly call them as? Metabolite, are you understanding? Hello, ma'am. Am I audible?
Yes, ma'am. You are
Okay.
audible.
Okay, ma'am. So metabolites are nothing but as we said, like these are the extra products which are being formed before the lead product is being formed. So the profiling is nothing but whenever we go through the mass spectroscopy techniques, into that sometimes it happens like we lead to the formation of different metabolites. Now we need to identify what these metabolites are. So when we go through the mass spectroscopy, we usually get different types of peaks. So these are the usually a parent peak, like into monkey hepatocytes and human hepatocyte. Hepatocytes are nothing but the cells which are present into the liver. If we isolate the complete liver, or we need to isolate some portion of the liver, that is some cells or tissues out of the liver, and then we pass through them after the procedure. When we pass through them through an mass spectroscopy, we get different types of peaks. So into these different types of peak, we usually get a basic peak in both the, while doing a comparison between a monkey hepatocytes and human hepatocytes, we get a comparison that we always get a parent peak. After a parent peak, we get different metabolite peaks. Okay. So sometime it happens like both the metabolite peaks are similar into the humans as well as into the animals. But sometimes it happens like metabolite peaks into the humans are different as well as into the animals are different. And these are the main aim where we can design a drug, or these are the main places how we can design the drug. Like these are the main factors for designing the drug. So the profiling of the drug. Next is characterization. Many times it happens like this drug identification is helpful in characterization. What is characterization? What drug does the molecule exactly look like? What is the molecular weight of the compound which we are finding, or compound which we are developing? Into that development of the compound, we usually get a metabolites as an intermediate. So through these intermediates, we can understand like where we need to make the changes so that we can make the drug more active. For example, we have already seen like many drugs, they have a certain ring structure. This is just an example. We have many ring structures present into the drug molecule. Substituting these ring molecules can make a drug active, or it can make a drug inactive. Now substitution of this drug making active and inactive can be understood only when we come to know the metabolic products, or what are the metabolites. For example, these are the raw material or raw drug. These raw drug can lead to n number of metabolite formation, and out of these n number of metabolite formed, we have to choose one metabolite which is having a very good activity, so that we can give a very good result out of the drug. The drug we have to make, that should have a very, what you can say, low side effects as well as very high activity. So these can be chosen, or these can be made with the help of the metabolite formation. Now again, these can be used to identify different types of metabolites, like which type of metabolite are formed. Same thing, like in humans, if the metabolites are formed of glucuronide metabolites, while in case of animals, they may be in the form of a sulfate metabolites. So we are finding a peak. These peaks are different also. But out of these peaks, which are the peak and into which category these peaks are formed, that can be even identified by the metabolite profiling or the characterization. Now, same thing, like typically this has been achieved by getting the retention time of the product, like what is the retention time, where does the peak fall, okay, what are the fragments that are helpful in authentication of the standard drug, whether the whatever products we have been getting, whatever metabolite we have been getting, that metabolite can be compared with the standard, and we can identify the range, what is the retention time, in which range of the mass spectroscopy these drugs have been falling. Now, this is just an example, like what are the different techniques of identification of metabolites. There are different techniques of identification of metabolites. It may be in the form of in silico technique, where we are using some softwares to identify which metabolites are formed. Like we are taking a sample and we are putting it into software onto a system. Considering the structure of the metabolite, we are putting it into a software, and then we can get the result. Like it will give a result which type of metabolites are formed. Either they are forming in phase 1 reaction, or these metabolites are coming under phase 2 reaction. On the contrary, we have another two different technique, that is in vitro technique and in vivo technique. Now, what do you mean by in vitro technique? It's like some portion of the body or some portion of the animals are being extracted or removed in the form of the cells, in form of tissues, in form of complete organ is been removed, and they have been tested externally. While in case of in vivo, directly we are introducing the raw material or sample in the body of the animal, and then we are testing the metabolites through the urine, feces, liver, intestine, lung, and kidney. And after this, in vivo and in vitro technique, we can identify the metabolites, and sometimes we can even quantify the metabolites. What is meant by quantification of metabolites? We can increase the number of metabolites, like ratio. Sometimes the metabolite, whatever is formed, these metabolites are coming in a very lower ratio. For example, if the raw material final product you are getting as 100%, sometimes these metabolites occur only in 10 to 20% ratio. So you can't study in only 10 to 20%. For studying these things, you have to quantify the metabolites, and for quantification also can be done through these metabolite products only. Now, what are the approaches of metabolite profiling? There are different approaches of metabolite profiling. It can be used for number one, metabolite identification. Which metabolites are formed? What are the different types of metabolite? What are the, as we studied in the previous slide, what were the molecular weight of the metabolite? What are the structure of metabolite? What are the different types of metabolites which can enter into phase 1 reaction or phase 2 reaction? Then we can even study the metabolic stability. Whatever metabolite is formed, for how long is it stable? Or do we have to introduce some stabilizing agent so that these metabolites can stay longer into our body? Next is rate and nature of the metabolite. At what rate these metabolites are formed? Like as soon as we have a medicine into our body, when the medicine enters into body, after entering, at what rate it is being metabolized, at what rate the metabolism is taking place, we can study it in vitro. And next is reactive metabolites. So as already discussed, like one raw material can lead to the production of n number of metabolites. So which is the reactive metabolite which is being formed into the metabolism process, that can be even identified by the metabolite profiling. Next is enzyme induction and enzyme inhibition can even be helpful with the help of metabolite profiling. Like if the metabolite formed are formed into the process, and these metabolites are very slowly being released out of the body. So at that time we have to introduce some enzyme induction, so that these drugs can be degraded easily and can be excreted out of the body. On the contrary, if the drug which are entered into the body, they have been converted into metabolites at a very faster rate, that time we can introduce the enzyme inhibition. So these enzyme inhibit the rate of the metabolism, and the drug can stay a longer period of time into our body. So reaction phenotype, like what type of reaction is going on into our body, what type of side effects these metabolites are giving to our body, that can be studied by the metabolite profiling only. Now, what are the different in silico technique? In silico technique is nothing but which we do onto a software. As I already said you, there are different types of software which are used to identify the metabolites. They are like Meta 2D printing, MetaSite, Silbil, or Meteor, or Metabol experts. So these are the softwares which can be used for studying the metabolic activity or the metabolites, whatever is formed. Now, what are the in vivo technique? In vivo technique, there are different techniques like number one is your S9 fraction. Now, what do you mean by S9 fraction? Here, you take a tissue fraction, okay, tissue fraction or a fragment or a cell, and these fractions can be used to study whatever metabolites have been formed externally. Externally, in the sense, whatever we are testing into the laboratory, whatever we are studying into the laboratory, that is known as an S9 fraction. Next is cytosolic fraction. What is cytosolic fraction? We have to take a simple single cell, or we can say we can take two cells, or we can take a chain of cells, and then we can study the activity over the single cell or the chain of cells. Okay, so we can understand like whatever metabolites are formed into a single cell, are they useful? Are they producing any side effects, or are they getting converted into the final products at a very fast rate? So that can be studied by the cytosolic fraction. Sorry. Okay, ma'am, am I audible? Hello.
Yes, ma'am. Yes, ma'am. You're
Okay.
audible.
Okay. So cell lines are nothing but these are majorly used to study different types of cancers. This is now becoming that metabolite identification has now becoming a very booming thing into a cancer treatment as well as cancer therapy, because cancerous cells, they usually get converted into some new what you can say new recombinant form, and they start regenerating itself into the body. So these cell line techniques, when we are testing any drug into the body, as soon as we inject any drug, they are getting converted into metabolites. So at what rate these metabolites are active? Sometimes are these metabolites itself active, like some reactive species, are they helpful in killing the cancer there itself? Or either they have to get converted into final product, and that final product are active over the cancer treatment. Next is liver slices. Again, some portion of the liver slices may be human liver or maybe rat liver. The liver samples or slices have been taken, and we can study the activity into the liver, because as we know, like whatever metabolism is taking place, the maximum like 90% of the body metabolism is taking place into the liver. So usually we prefer taking the human liver, or we prefer taking the hepatocytes and liver slices and studying in vitro, like into the laboratory, cutting a piece of a liver or taking a tissue out of a liver or taking a cell out of a liver and putting that liver cells into the testing in laboratory is a very helpful technique for understanding the different types of metabolites. Now, we have to, in if we are studying an in vitro technique or in vivo technique, whatever it is, we have to extract the particular cells. So for extraction of the particular cells, there are different types of technique for extraction. We can use, number one is protein precipitation. Number two is liquid extraction, and number three is solid phase extraction. Now, what is protein precipitation? Whatever proteins are being present into the sample, that proteins are reacting with the metabolites and they get settled down, and we get a plain supernatant liquid. So the protein reacts with the metabolite and we can easily find the metabolites whatever is formed. While second is the liquid extraction. Whatever product you or drug you have injected into the body, the sample you have taken externally, maybe in the form of your serum or maybe in the form of urine, maybe in the form of saliva, that sample has to be mixed with some organic solvent. So when it is mixed with the organic solvent, they form different layers. As you can see on the screen, they form different layers, and these different layers are helpful in separation of your metabolites. Now, solid phase extraction sometimes happens like these metabolites, they get converted into some solid products, and automatically they sediment down, and the supernatant we get a clear liquid. So these solid phase can be easily extracted into the solid phase extraction. Now, there are different analytical techniques also for identification of metabolites. We can identify the metabolites using the mass spectrometry, HPLC, LC, GC, MS, FTIR, Raman spectroscopy. So these are the different types of techniques which are used for identification, okay, for the identification of your metabolites. Now, for analytical technique, we usually require a presence or preparation of your sample. This is the first and the foremost and very important step for preparation of the sample. If the sample is not properly formed, or if there is some hindrance in the preparation of the sample, these can become an hindrance into the spoilage of the analytical instrument itself, because the instrument whatever you are using, that is LCMS, GC, HPLC, IR spectroscopy, these are have very costly and expensive units. So if you don't prepare a sample properly, if there is some mistake in preparing a solvent or preparing a sample, and that sample can get hindered or can get attached, or it can destroy the analytical instrument whatever you are working with. Okay. So next is like sample preparation includes all the procedures and operations applied for sample prior to the analysis. This is mainly implemented to stabilize the sample, to remove the contaminants out of the sample, to enrich the sample. Like whatever sample you have to enrich the sample. Enrich in the sense, for example, your sample may be in the form of liver cells, or your sample can be in the form of plant extract. So these extracts sometimes have extra contaminants with them. So removing these extra contaminants and making the sample enrich is a very important and the first and foremost step which is involved in the analytical techniques for identification of the metabolite. So we can avoid fouling the mass spectrometry, whatever the spectrometer we get. Suppose in case if we do not remove the contaminants, what will happen? We will get the peak of the what you can say, we will get the peak onto the mass spectra, but these peaks may be of your contaminants. Many time it happens like we get the peak of contaminants, and that can spoil whatever product you are getting, or that can give you an idea about the false metabolite, even though it is not a metabolite, but still you are getting a peak which indicate like you have a contaminant. So preparation of the sample is a very important step into the analytical technique. Next is choice of sample. It should be very extremely important for metabolomic study. Like an ideal sample preparation includes, it should be non-selective. It should have a depth coverage of the metabolite, whatever metabolite you are expecting to get. Like we have an idea, we are extracting from a plant. Into that plant we have an idea like what are the different metabolites it may be having, or what can be the expected structure which we can find into it. So considering those, we have to select a sample, or we have to prepare in such a way the solvent should be non-selective into which we are mixing, so that the metabolite whatever they are formed, these are formed in a very simple and a fast way, and our metabolites are not destroyed during the high throughput screening, or during the analytical technique. Whatever metabolites we are getting, these should not be destroyed. Next is, these should be reproducible. Reproducible in the sense, suppose if I'm carrying an experiment into my lab, taking the plant extract, and if I get certain peaks, so I should be reproduced, it should be reproducible. I should be reproducible it into in front of the other like suppose my seniors or my principal investigator, or the head of the project whatever we are doing. So we should be able to reproduce the results in front of them. These were the results which I got when I was doing into my lab, and the same if I am performing into the university, the same results should be reproducible. Now metabolic quenching should also be it should be taken care of. Like quenching usually is nothing but a reduction of excited state. Sometimes it happens like there are different metabolites formed, so these metabolites have their own excited state, so we have to reduce the metabolic quenching so that it should not interfere into our analytical technique. Now same sample preparation, you have to collect the sample first, you have to reduce the quenching with the help of methanol. Quenching is nothing but reducing the excited state. You can do it with liquid nitrogen or cold methanol. Then you have to extract all the intracellular metabolites, and you have to concentrate it. You are concentrating either with under vacuum, evaporation under vacuum, or you are concentrating it with lyophilization technique, or a simple extraction technique. Now, sample preparation method. There are two different sample preparation method. Number one is a dilute and shoot method, and number two is a solvent precipitation. What is a dilute and shoot method? Whatever sample you are taking, for example, if you are taking a plant extract, this is the sample what you are putting. So you have to convert the sample in such a way that as soon as the sample is being prepared, for example, the sample is coming into a supernatant liquid, the supernatant liquid is directly being injected into your LCMS apparatus. So that is known as an dilute and shoot. Whatever sample you are diluting, that diluted sample is directly being sucked off by the LCMS techniques, or whatever analytical method you are using. That is this method is known as a dilute and shoot method. Next is a solvent precipitation. Here we are diluting the sample. On the contrary, solvent precipitation, we are precipitating whatever the sample is. Sample, we are taking the sample, we are diluting it with some solvent, and the solvent react with our sample, and they get precipitated. So these precipitates are used for the testing further. Okay, in case of dilute and shoot, directly that material is been entering into the analytical machine, maybe LCMS or GC. While on solvent precipitation, initially we are taking the sample in the form of plasma or in the form of biofluids, in the form of serum. These are the preferred samples which we are taking, and then we are automatically a protein is being precipitated with the help of the organic solvent. These precipitates can be used further for the sampling techniques. Now, what are the liquid extraction? This we have already studied in the prior slide only. Liquid extraction, like usually we this is a very common sample preparation method, liquid extraction. Into this liquids in the form of cerebrospinal fluid, sample in the form of cerebrospinal fluid is been taken, and that can be used with the help of, or that can be you can mix it with some polar solvent, or you can even combine it with the aqueous solution, that is water. We can combine it with ethanol, dichloromethane, etc. And then you have to go through first, excuse me, someone has kept their mic open. Can you please put it off?
Yeah, ma'am, I have muted them, sorry.
Okay. So whatever sample you are taking into liquid extraction technique, that has to be mixed. Sample has to be mixed with initially the sample has to be mixed with the solvent. That may be sometimes a polar solvent, or that may be a non-polar solvent, depending upon whatever sample you are taking. The sample has to be mixed with the solvent, and these solvents are after mixing the solvent, you have to put it through a vortex. Okay, vortex is nothing but as you get a cyclone, pura circle, okay, they get whirled or they mix into a vortex. After mixing, you can again pass it through the centrifuge. Centrifugation, once you do the centrifugation, the layers automatically get separated. We find two different categories of layers, and that indicates like we have separated the solvent as well as our metabolite. Our metabolite reacts with the solvent, and they get separated into two different phases. Same protein precipitation, nothing but the sample are taken and they are mixed with the organic solvent. They get precipitated with the addition of acetonitrile. Whenever we add acetonitrile or acetonitrile methyl alcohol, whenever we add it, and we centrifuge it, automatically the proteins which are present into our sample, maybe the plasma sample, maybe the blood sample, maybe the serum sample, they get precipitated at the lower end, and we can easily extract those sample. Next is in vivo sampling. For the in vivo techniques, we are doing sampling nowadays. It's become a very important task, and people are preferring in vivo sampling technique compared to the in vivo sampling techniques, because in vitro is whatever sample you are directly injecting into the rat or mice, or you are injecting into the human liver cells, or you are injecting into the human liver tissues, where automatically the metabolites are formed in vivo in the body itself, and you can remove those metabolites without getting destroyed. In vitro technique, there are some drawbacks, like whenever you are working with the in vitro technique, sometimes the metabolite get degraded when it comes in contact with or exposure to oxygen. Whenever the metabolite comes in contact with exposure to oxygen, they get destructed. But here, in case of in vivo technique, the destruction or coming in contact with the external oxygen or environment is very rare. So people are now preferring the in vivo sampling technique. There are two different techniques. Number one is microdialysis, and number two is solid phase micro extraction. Okay, so what is microdialysis? Microdialysis is nothing but sampling of only, this is done only for lower molecular compounds. Okay, lower molecular compounds that are formed in the blood and the tissue. Microdialysis technique into this, like blood and tissue samples are taken. For example, my sample that is being injected into my blood. After injecting into my blood, we have to wait for a certain period of time so that it gets converted, or so that it gets metabolized. After metabolizing, we have to extract the sample from the blood and the tissue, and same thing as we do in dialysis. What we do in dialysis, the blood is being removed and it is being put into the dialysis machine. All the not so important or the constituents which are not required from the blood have been sorted out, and the fresh cleaned blood has been passed into the body. Same way, it's a similar technique like micro dialysis. We have a dialysis apparatus which is kept externally, and if this is a human body, into the human body, as soon as we inject the medicine, we have to wait for a certain period of time so that the metabolites are formed. When the metabolites are formed, we have to pass our blood through the microdialysis apparatus. As soon as we pass through the microdialysis apparatus, the metabolites are separated and the clean blood has been entered back into the body. This is one technique which is used for extraction or sampling technique for microdialysis. And number two is the solid phase extraction. So solid phase micro extraction, it is the direct sampling method where the mouse blood is been taken when the mouse is awake. Okay, when the mouse is awake, in the sense, same thing, we are injecting the drug into the body of the mouse or a rat, and then we are I'm sorry. In vivo in vivo sorry, in vivo solid phase micro extraction, the direct circulating sample has been taken from the circulating blood mouse. That is when the mouse is awake. Sometimes in case of in vitro, we have to take the sample of the metabolites directly from the I'm sorry. I am I audible?
Ma'am, you are audible now.
Okay. So in vivo solid phase is like sorry, I was like sometimes it happens like in case of in vitro technique, the we have to kill the organism or we have to sacrifice the organism, and then we have to extract their liver or the tissues or the cells from the liver, and then we have to do the sampling. While in case of in vivo technique, when the mouse is awake, that time only we can take the circulating blood of the mouse and we can extract the hydrophilic and the hydrophobic metabolites, so that it is easy for us to put it into dialysis. Same way, we have to put it into dialysis and we can put that sample through the GCMS or LCMS, and we can get the different metabolite structures, whatever is being observed into our sample. Now, can we perform the metabolite characterization in vivo sample? Yes, sometimes we get a question like whatever we dealt with, like it's easy for us to sacrifice a mice, take the liver out of the mice, and we can study those hepatocytes very easily. So can we perform the metabolite characterization study in vivo? Yes, it is possible. We can perform the metabolite characterization in vivo by taking the samples of plasma, urine, fecal homogenate, or various homogenates which have been formed. And sometimes we can even radio label those metabolites. Radio label those metabolites in the sense, whatever medication we are putting into the body, that medication has to be labeled. Radio labeled in the sense just to identify whether they have formed a metabolite or not. In case, example, we are putting C14, at the end we should get a product like C14 only, but we are getting certain metabolites into our body which gives you C13, which gives you C12. So these are nothing but metabolites which are radio labeled and which can be easily identified by the in vivo techniques only. So it's very easy nowadays it's become very easy to categorize, like characterize the metabolites by performing the in vivo studies by using the samples of plasma, urine, as well as different homogenate. Now next is dried blood spot. Again, this is an emerging technique. This is not yet developed, but still they are doing a study over the dried blood spot. This study actually came after one child who had a disorder after the birth itself. Like when the baby was born, they came across many inborn errors into the metabolism. Like the baby had many inborn metabolic problems. So to understand these metabolic problem, what they did is like they have extracted few blood samples from the baby, and they have kept it over a paper, filter paper, and soaked over the paper, and then they have studied those soaked blood spots, analyzed those blood spots, and after analysis they come across many metabolites which are not formed into the normal newborn baby. For example, normal newborn examples. Okay, for a normal newborn baby, we find 1, 2, 3, 4 metabolites are formed into the normal newborn baby, but a baby with a defect of a metabolic problem, you usually find 1, 1.1, 1.2, 1.3. These are the additional metabolites which have been formed, and they can be identified with the help of dried blood spot technique. Now, what is dried blood spot technique? You have to take a sample of blood of a normal baby as well as a defective metabolic problem baby. So you have to take those blood spots, you have to soak it on a paper. After soaking it on a paper, you have to dry it for several hours, and then those spots can be extracted with the help of samples like methanol analysis, or you can even go through again GC or MS analysis are the bestest thing to identify whatever metabolites you have found. So with the help of these metabolites is again very easy for us to understand that these babies were having metabolic problem initially, like when the baby was in the third month, or when the baby was in the fourth month of pregnancy, sixth month of pregnancy, and that time itself we can rectify whatever problem we were having. Suppose, are you getting my point? Hello? Can
Yes,
you hear
ma'am.
me?
Yes,
Okay.
ma'am.
Okay, so this is dried blood spot technique. Next is laser micro dissection. We are dissecting or splicing or removing some of the cells which are of interest. For example, the tissue or the organ or a cell. We are isolating it with the help of laser technique. Okay. After isolation or dissection, these have been widely used for studying like genomics, for transcriptomics, proteomics studies. They have been widely used. These dissected samples, maybe tissue, maybe cell, or maybe organ, can be dissected with the help of laser technique. Means we need not cut open our body and remove the liver portion or cell portion. We can just pass on a laser into the body, and with the help of the laser, that can be extracted very easily, a painless manner. Only a minute cut is required, and through which you can just extract the cells with the syringes. Now, a variety of the samples can be performed with laser micro dissection technique, like blood smears, cytological preparation, cell culture, and tissue techniques. Now, laser LMDs is metabolics involves the sample collection. What are the different steps involved here? First is sample collection, fixation, embedding, sectioning, and harvesting. What is it? Like initially, we have given a small cut over the body of a rat. For example, this is a rat liver. We are giving a minute cut in the abdomen. After giving a cut on an abdomen, we are just putting a syringe into the body of the rat, and we are extracting the cell samples. After extracting the cell sample, that is number one step, that is collection. After collection, we are fixing the cell samples, usually with the help of some amount of organic solvent. While fixing with chemicals, we have to be very careful. We have to fix it over the slide. While fixing it, we have to be very careful because sometimes it happens that chemicals may react and react with our tissues and cells, and your cells may die. So you have to be very careful while choosing the chemicals. Then you have to embed it. You have to embed with the help of buffered saline or phosphate, cold phosphate solution. We usually don't prefer paraffin because that can cause the delocalization and loss of metabolites. And last but not the least is you have to harvest these. Harvest means you have to cultivate more amount of metabolite, whatever metabolites are formed, you have to harvest it, you have to preserve these metabolites for the future studies. Repeatedly removal of the same cells, harming the animal for the same procedure, it becomes a difficult task, because ethical clearance and ethical committees have become very stringent in this regards, and repeated harming the animals has become a difficult task. So whatever samples you have been removed, you have to regenerate those samples, and you have to preserve and harvest those samples for the further studies. Next is microwave assisted as extraction. Whatever metabolites you have been formed, that can be extracted with the help of microwave. But here you have to take care of the optimum time and the temperature. Whatever time you are microwaving the sample, or whatever power, at what temperature, at what power you are keeping it, that has to be very important, because sometimes it happens like if you keep the temperature higher, or if you keep it for a longer period of time, the sample may get degraded. So we should always use the optimum time and temperature, so that efficiently we can get more amount of sample or more amount of metabolites, whatever is formed into our sample. Next is ultrasound assisted extraction. This is the basic technique which is used in almost many laboratories of metabolites, where we can just a simple, I hope you all might have seen what is an ultrasound. Ultrasound is nothing but an equipment where you keep your samples into it, and with the help of sonic radiations, the product are being extracted. For example, if you keep a sample into it, the sample won't get destroyed because of these ultrasonic waves. They just get extracted. Whatever sample has the metabolite, it gets extracted and it gets settled. So this decreases your extraction time and increases your metabolite concentration. What ultrasound with the help of ultrasound technique, you get more amount of product whatever you are expecting, and that too at a very cheaper rate, or it is, or we can say like the cost of the production through the ultrasound technique is very very low. So we can use different types of products like leaf or plant extracts can be easily been extracted with the help of ultrasound. Usually we use ultrasound for plant extraction only, and this is a very common technique which is used in almost all the laboratories for extraction of different types of metabolites. Enzyme assisted extraction is nothing but here you are using certain enzymes which are helpful in extraction. Like for example, if you put your sample, along with the sample you are pouring some enzymes. What these enzymes are doing, these enzymes are helpful in and these enzymes enter into your sample and they help in extraction of whatever metabolites you require at that particular phase into that particular time. So extraction by enzyme assistant, again it's a developing technique. Sometimes it can disrupt your cell also. Sometimes it can destroy your cells also. Enzymes not necessary can be only healthy. Sometimes it can be destructive also to the cell. So you have to be very careful in both the ways. Like enzyme assisted is good. It can disrupt the cell, enter inside, remove whatever metabolite you require, and then you can easily extract the metabolites. But on the contrary, sometimes it happens like it enters into the cell and destroyed the cell itself. So you, when once the cell is getting destroyed, you won't get any of the metabolites out of it. Now, in vivo metabolite identification, how are you identifying the metabolites? There are different techniques. Usually, we prefer the technique that is LCMS, that is liquid chromatography, as well as high resolution mass spectroscopy. These are the major important thing which are useful for identification of almost all type of metabolites. Maximum metabolite studies have been done with the help of LCMS only. Okay, or LCHRMS, that is high resolution mass spectroscopy. These have been majorly used for studying your ADME and different types of, like, what is the rate of absorption, distribution, metabolism, excretion, at what rate your metabolites are formed, at what peak you are getting into the mass spectroscopy, that can be easily identified. Okay, now there are a few protocols which you have to follow in case of LCMS analysis. Like you usually prefer the sample maybe in the form of plasma or serum, and you have to keep it over the wet ice. After that, you have to place it into a 96 well plate, and you have to add acetonitrate, that is 40 ml of acid, 400 liter of acetonitrate. You have to vortex it. Vortex is nothing but a whirling or mixing movement. You have to vortex it, then you have to add again acetonitrate, then you have to centrifuge it so that the product get easily separated. When you go it into a stepwise manner, it becomes very easy for you all to separate the samples very easily. Now repeat the step 1 to 5 again till you get what optimum amount of extract you need, what optimum amount of metabolites you need. Till that time you have to repeat the 1 to 5 step. After that, you have to precipitate the sample, whatever sample you have taken, you have to precipitate it, and you have to centrifuge it again. You have to remove the filter plates and seal the 96 well, and avoid the immediate storage and avoid the samples analysis of the sample like immediately once you complete the procedure like centrifugation. After you complete the centrifugation, you should never take the sample for analysis. You should always store it for a certain period of time, and then you have to analyze the sample. For tissue samples, again, you have to take a frozen tissue. For example, if it is human tissue, or it is a rat tissue, or it may be a tissue from a plant extract, you have to take a frozen tissue. You have to keep it on a dry ice. Then you have to mix again methanol at the ratio of 500 liter solvent per tissue. Optional is like in case of the metabolite is very, very dense, then you have to dilute it again. In case, for example, the sample, if it is of leaf, you can extract if it is of leaf, you can extract the sample very easily from a leaf. But If it is, if you have to extract the sample from the bark, it becomes very difficult, because whatever extract you got from the bark, that would be thicker into consistency. At that time, you have to dilute it again with the sample. After diluting only, you can further proceed to the homogenization step. Homogenization is separation. Homogenization, again, you can skip, if you don't have a homogenizer, you can optional, like you can put that thing into a mortar and pestle with the liquid nitrogen, and you can homogenize it. Then again, you have to centrifuge and collect the sample. Again, there is an optional step, is like filter the sample and take the aliquot. So these are again, next is a protocol for HRMS. Same thing is repeated over there also. Next is in vitro technique. Now, what are the bestest way of in vitro technique? In vitro technique is cryopreserved hepatocytes. Like, suppose in case if the rat is died in front of your eye while you were doing experimentation, at that time instantly the rat died. So you have a liver, or you have all the organs working for a few minutes to few hours. In that second, you can remove the liver of the rat or mice, and you can cryopreserve them into liquid nitrogen, so that you can utilize these hepatocytes for their further studies. Instead of wasting the animal, instead of throwing away the dead animal, sometimes we remove the organs. Like in humans, as soon as the person die, we remove their eye are donated. Then very different parts of the bodies are donated. Same way, as soon as the rat or mice die in front of you, ऐसा नहीं कि it's already died 2 days back, you are removing a liver out of it. No, it's not possible. As soon as the rat is died in front of your eye, you just have to take that eye, you have to dissect it body, you have to remove the liver out of it, and you can cryopreserve it. And this cryopreserved can be used for in vitro technique. So this cryopreserved thing, what it can be used for? It can be used to study the different metabolites which have been formed by the hydrolysis process, reduction, oxidation, or the conjugation. Hmm. Next is human liver microsome and rat liver microscope. Okay. Now, there are two techniques in vitro. One is a microsomal technique and another is an hepatocyte technique. What we have studied here was an hepatocyte. In hepatocyte, you are taking the entire intact liver. पूरा का पूरा liver ही आप इस्तेमाल कर रहे हो, और ये पूरा liver ही आप study के लिए you can use it. But in case of microsomal technique, you can use the inside portion of the liver. Inside portion of the liver, in the sense, you can remove a single cell and you can study over that cell. You can remove a single tissue and then you can study over the single tissue. Okay. Now, human liver microsome. What is a human liver microsome? It is nothing but you have to take the hepatocytic cells from the endoplasmic reticulum, and it is being obtained again by centrifugation. You have to take a portion of the human liver or slices of the liver or liver cell line, and you have to homogenate it. Homogenate मतलब छोटा छोटा pieces में उसको cut करना. You just have to break down into the smaller pieces. These human liver are nothing but a good source of different types of enzymes like cytochrome P40, flavin monooxygenase, carboxylic esterase, and a number of enzymes have been formed into the human liver microsomes. So therefore, these are frequently used for in vitro modeling, that is metabolic profiling or metabolic identification or drug interaction studies. What drug interaction studies are there? Drug interaction studies, in the sense, whenever you take a liver sample, for example, you take a liver cell or liver tissue. If this is a liver tissue, you have a number of enzymes present into it. As soon as you take your sample and you pour over this, they start reacting with one another, and you lead to the formation of different types of metabolites, or you have different drug interaction are being formed. Different drug interaction, in the sense, whatever the drug has reacted, it is easily being reflected. Either the drug is good enough to what you can say, good enough to solve the disease what you were having, or the drug is having many side effects that can be clearly identified with the human liver microsome. These can be even used to study the inter-individual variation. Inter-individual variation, in the sense, this can be used to study the difference into the human, like male as well as female liver microsomal enzymes. In the sense, if I am a female, my liver may contain different microsomal enzymes, and if there is someone male into the study, his liver may contain some microsomes which are different. So for testing a drug, we can take the liver sample of a male as well as female, and we can test it so that we can get an inter-individual relationship between what metabolites are being formed into the males as well as into the females. Apart from these liver samples, you can even take the different human samples in the form of intestine, kidney or liver, and they can be used for extrahepatic metabolic studies. Apart from liver, agar koi metabolism ho raha hai, so that metabolism is usually occurring into the intestine, liver and kidney, and that can be again very easily identified. Okay. Next is additionally the gender, as I already discussed, like gender specific microscomes can also be studied through this technique. In drug discovery process, again, it's used for metabolic identification. It is used to study the interspecies difference. What is the difference between interspecies? As we have studied in the first slide, what metabolites are formed into the monkey and what metabolites are formed into the human. So it's very useful to differentiate between the interspecies metabolism. Now, next is it is useful in the prediction of in vivo clearance also. Clearance in the sense whatever drug you have intaken, that drug either it is being excreted out of your body, or it is being bound to some protein, or it is still present in your body in some other form. So that can even be studied through this HLM technique only. Now, reaction phenotypic and metabolic pathways can also be identified. Like in biochemistry, you might have studied different metabolic pathways. Those metabolic pathways, their problems in the metabolic pathway, what enzymes are required, what energy they are releasing, either in the form of NAD, ATP. So those also can be studied through these metabolites, whatever is formed. Next is what is the advantage of HLM? It is very easy to use. It's low cost. It's best characterized in vitro model. It's the bestest technique. Like rat ka apne sorry, human cells you have taken, human liver cells you have taken, you are comparing, you are getting the result out of it. So it's the best thing which is giving you an idea of what the drug is going to do to your body, or how many metabolites are formed into your body. That can be easily identified by the HLM technique. This is appropriate for studying the inter-individual and population-based variation. Long-term storage is also possible with the liver cells. Whatever liver cells you have extracted, not necessary like you have to use it today itself. You can use it for further studies also. You can keep it or you can preserve it, and then you can later use it because these cells won't die so easily. So it provides the qualitative analysis of in vitro drug metabolism. It's a convenient tool for high throughput screening. It's an appropriate for lead compound optimization as well as drug interaction studies. Whatever interaction is going to be taking place into your body, that can be easily studied through the HML technique. Now, there are some disadvantages, like it is inappropriate for quantitative estimation. Quantitative estimation matlab is not giving you an idea how much quantity of metabolites are formed. For example, if you have injected a 500 mg you have taken a 500 mg tablet, and you can't say ki out of 500 mg tablet there were 250 mg metabolites are formed. So this is not a quantitative way, it's only qualitative way. What quality of metabolites are formed, what are the different metabolites which are being formed, that can be identified. What are the molecular weight of metabolites, even that can be formed, molecular weight. But they can't give you the quantity of how much biotransformation is being taken place, or how much metabolism is still yet to take place. That can't be identified up till now. Another drawback, it is very difficult for assessment of fractions of drug bound to the plasma protein. As I said you, like sometimes it happens like some metabolites, some drugs, as soon as we intake into our body, absorption, distribution, then is metabolism. Into distribution, sometimes it happens like drugs, they go and they bound to the plasma protein. So when they bound to the plasma protein, they are unable to get converted into metabolites. So here it's difficult to identify like how many drug molecule is being converted into plasma protein or bounded to the plasma protein, and how much amount of metabolites have been formed out of the total drug what we have taken. Next is liver microsomal preparation protocol. It's not necessary, I think. We are running out of time also. Next is rat liver microsome. What is rat liver microsome? Instead of human liver microsome, we started taking rat liver microsomes also. Livers have been harvested from Bristol rats, as well as brown rats, as well as bobwhite quill rats. So these rats are usually used for the microsomal studies. They have been anesthetized, and after giving anesthesia, they have been cut from the abdomen, and the liver was removed. They have been weighed. The tissue is being rinsed thoroughly with NaCl and immediately frozen with the help of liquid nitrogen. They have been frozen and they have been kept so that they should not get destroyed, and we can use it repeatedly. Now perfusion was continued until the liver appear blenched. What do you mean by blenched? Is like completely all the extra ingredients, whatever the blood, whatever the liver was having, that has been removed off, and with the help of liquid nitrogen. Now perfused tissues with the liquid nitrogen. The frozen sample were kept and stored at -80 degrees Celsius until the further process. Like whenever you require, you have to just remove the frozen sample, liver sample, and you can do the microsomal testing over it. So there are different types of test solutions which can be used. All the formulation consists of diphenoxyquinone. These are some buffer solutions have been added, and then they have been homogenized. The liver microsomal preparations, these are usually centrifuged, spliced or minced into the smaller pieces, and they have been mixed with buffer. After mixing with buffer, we are again centrifugation has been done. After centrifugation, the supernatant liquid is being transferred, and whatever the metabolites are formed, they have been settled down. So this suspension and removal processes until you get the desired amount of sample, whatever is required. So these microsomes, they can be incubated at 37 degrees Celsius for 60 minutes. So residue determination is not required, I think. Now, will I get a quantitative information of the metabolite studies? No, I can never get a metabolic study, a quantitative study into the metabolites. Just as I said you, like unless we put a radio labeled drug into our body, we are never going to get a quantitative study. Yes, if you are going to put a radio label substance into your body, that radio label substance is going to show you. For example, if I am putting 500 mg of 500 mg of C14, so at the end, how many metabolites of C14 are formed, that can be easily calculated or counted. Why? Because these are radio labeled. So radio labeled, they can easily be identified, like 250 have been identified, 150 have been identified. So unless it is a radio labeled, you cannot quantify it, because it's very difficult, as I already said, like some are protein bound, some to quantify. Again, there's an example like only one structure, if you take a basic structure, one structure may be having different types of metabolites. Okay, these metabolites may be in the form of, they'll give you different peaks. If you put on a mass spectrometry, they will give you different peaks. So they may be in the form of, see, if you see the peak height here, the ortho and the para, it's almost similar, but the meta structure is different. So we cannot identify which metabolite was higher into the production and which metabolite was lower. So these are the different types of metabolite, and so we cannot identify these metabolite quantitatively, but qualitatively we can identify. Qualitative is like, we can after getting the peaks, we can identify this is the ortho peak, this is meta peak, this is your para peak. So these can be easily identified. Okay, I think here I complete my topic. So if you have any doubts, you can ask me the doubts.
Ma'am, thank you. Thank you very much for your nice presentation. Thank you so much, ma'am.
Thank you, ma'am.
Almost all the topic you have covered, and very fastly
Yeah, ma'am.
in time.
Because I've been said like we have
In
only
time.
1 hour of time, so I went it a bit faster.
Not like that. Not like that. The thing here is minimum we consider is to 1 hour. So we have some queries from students, and
Yeah, ma'am.
now I request Rihanna ma'am to please question it. Ma'am, do you have any questions? From one of the participants, we got information that very informative session. Just a minute, ma'am.
Yeah, ma'am.
Hi, ma'am. Participants, do you have any questions? Please post in a chat box.
Sample preparation also, as we are from analysis department. Thank you so much, ma'am, for covering all these topics like metabolite identification, characterization, the difference, and reactive from macrosomes, and the way in which we take this dried mix plot. All these things we have covered. Thank you so much, and almost most of the fifth unit of MBAP we have covered, I think.
Thank you very much, ma'am. I really thank all the team and the organizer team, especially you, Dr. Sunita ma'am, as well as Rihanna ma'am, for giving me an opportunity, ma'am. Thank you very much.
Really, I thank Rihanna ma'am also to introduce you.
Thank you.
She told about your name when we talk, when we are when we want to organize a meeting. So
Yeah, ma'am.
initially, thank you so much, Rihanna garu. And at the same time, ma'am, have you done any time with this type of work in your lab?
Ma'am, I have been going through some molecular docking studies. So molecular docking studies, again, very, very important. And nowadays it's having like a too much scope into to understand the drug at a molecular level. Where these drugs are going and binding to your receptor, it makes us very clear to understand in which way or in which part of the body the drug is going to go attack, and they are going to produce which type of orientation. So molecular docking has been helping in identification of the particular receptors and the way of action. So we have been going through, specifically me, I am going through some molecular docking studies only.
So
So it's,
you mean to say through software
yeah, ma'am.
in silico studies you are doing,
Yeah,
right?
yes, we are going through software. The things like initially it was very difficult for us to understand the structure as well as the receptor. Like we just knew that this is the structure and these are the receptors. But are these structures, are our drug molecule directly going and attaching to that particular receptor? That was a very difficult task to understand. But now through the software, it has become so easy. We can go with a particular orientation inside the structure and we can identify at which particular point they are binding. Are they going to produce the desired effect, what we are saying? Initially we just used to extract the plants, we just used to extract the material, and then we just used to isolate the compound. These isolated compound, are they really active, and to what extent they are active, we can study through the docking study. So through the software we get like numerous compounds are active, and still there are like numerous study which can be done over single receptor. Considering only a single receptor, you can go through a numerous structural changes where new molecules of the drugs can be easily developed.
Yeah. This is the one type of source what we have nowadays for research.
Yeah, yes, ma'am. Yes.
To do as per the software and all.
Yeah, ma'am.
Anyway, thank you for the information, ma'am, and thank you so much for
Thank you,
presenting such a nice way. Thank you. Thank you, one and all. Dear participants, if you have any queries, just moment, ma'am.
Just a second. I think there's a
Ma'am,
question.
which qualitative method is more efficient for metabolite profiling?
See, now what
Qualitative.
qualitative, see, everything is qualitative only because we don't have anything quantitative for metabolite profiling. Quantitative, as I said, is we don't have quantitative identification. हम identify ही नहीं कर पाएंगे, quantity ही नहीं पता चल रहा है ना. When you have a drug, when you take a drug, that drug is entering into your body, that is getting absorbed, distributed, and metabolized. How much is metabolized, that we cannot quantify. We are just getting on to quality. Which quality of metabolite is formed? Either it is entering into phase 1 metabolic reaction or it is entering into phase 2 reaction. That can only be quantified, with the help of which we can stop certain disorders to take place. For example, जैसे हमको पता चला one of the metabolite is which is being formed, this is causing rashes to our body. Example, this is causing rashes to our body. What can we do? We can stop that metabolite there itself. How are we going to stop? We are going to add one additional agent into our drug so that it can block that particular metabolite, and rest all the drug is being useful for our body.
So which qualitative method? Mean to say only MS?
Yeah, majorly we use MS only, ma'am.
MS. So
Mass
without
spectroscopy.
having this MS, probably we cannot go with this metabolite.
Yes, ma'am. We have to have MS only.
So, dear participants, I hope you got the answer. Okay. Thank you so much, ma'am. And Rihanna, ma'am, do you want to speak? Yeah. Please mute yourself. Unmute. Yeah.
Thank you so much, madam, for your very, very informative session. And it is
Thank
very
you, ma'am.
clearly understood by our participants now.
Thank you very much, ma'am. My pleasure.
Yeah, on behalf of our principal also, we thank once again. Thank you so much.
Okay. Bye, ma'am.
Yeah, yeah, thank you so much, ma'am. And in future, if we have any questions, we will approach.
Sure, definitely, ma'am. Sure, definitely you can.
Yeah, thank you.
Okay, ma'am.
Yes, dear participants, please complete the Google form, whatever you're going to have through WhatsApp group. Thank you so much for attending the seminar. I hope we can be.
Can I end the meeting, ma'am?
Yeah, ma'am.